What's going on, guys? Salman Ahmed here, back with another video from SalMed, and in this week's video we're going to be showing you a week in the life of working in an undergraduate research lab. Let's go. So for just a quick overview for this week's video, we're going to be doing a microdissection of the Drosophila melanogaster, or commonly known as the fruit fly. And I know you're probably wondering, why are you researching and dissecting fruit flies? Fruit flies are a great model organism, which means that they hold a lot of the similar biochemical pathways that also run within humans. They're also a lot easier than researching with bigger organisms such as rats or mice. Our lab specifically looks at the development of synapses between neurons, or in other words, brain cells. For those of you that don't know, the synapse is the area between the brain cells where communication takes place. So information can be transferred from one brain cell or one neuron to the next. So if we can get a better understanding of how normal synapse development occurs, then if something goes wrong, such as a neurodegenerative disease, then we can understand that what's not working here, what's the problem, why is this happening? So if there's a problem and if we know how it should normally work, then we're gonna know what's happening when it's improperly working as well. So as I kind of alluded to earlier, we're gonna be doing a microdissection of the central nervous system, specifically of the fruit fly. So this is typically a 3-day experiment, with the first day being today. So we're gonna go ahead and get started and head to the lab, and I'll see you guys there. Alright guys, so I'm going to go ahead and give you a quick tour of the lab and show you guys some of the stuff that we work with. So this is one of our 2 microscopes that we use to dissect the flies. The other microscope is right here. It's covered right now because I usually prefer to use this one. Over here is our fly incubator, so this is where we keep our different genotypes of flies that we're going to experiment with. So my box is right here, and I'm going to be picking out 2 of the genotypes to dissect today. This is our workstation where we prepare our experiments and do different washings with. So you guys will be able to see some of that stuff later. This is like another general work area where we keep our stuff. If we need to do any writings, we do it over here. And over here is our immunofluorescence microscope. So we do more fly work here, specifically looking at the samples that we dissect and stain. And over here is our other fly incubator, which we keep more different genotypes that we experiment with throughout the year. So there are 2 main incubators. This one holds various different genotypes that we use, and then the incubator over there focuses more on stuff that we're focusing on for the week. We're going to start off the experiment with preparing the solutions that we're going to use today. So we're going to go ahead and get started. This right here is formaldehyde solution, so it's one of the few things that we work with in this lab. We actually have to wear gloves while working with it because it is a known carcinogen. So if it enters our bloodstream through a break in our skin, then it could lead to cancer. So while working with it, we take all the necessary precautions, which in this case is wearing gloves. Alright guys, so we have everything ready and set up to dissect, and we have our two pliers, you can call them, to actually do the dissection. I hold one in each hand, and we usually get started with the control group in this case, which would be this set of flies, and then we pull out the larvae. Usually put out 6 of them, and we put them on that glass plate over there, and we usually start our dissection. Usually takes about 20 minutes to do both. So I'm gonna go ahead and get started, and I'll see you guys in a bit.
Twenty minutes later.
Hey, what's going on, guys? So just finished the dissection part of the experiment. I have about 8 minutes before my biochemistry class starts. I have the luxury of doing my class in the lab since everything is online. I'll be finishing the rest of today's experiment after class, and I'll be finishing the rest of the other 2 experiments between Tuesday and Wednesday. So I'll be taking you guys along with me on those days as well. So right now we have class, so I'll see you guys there.
Refers to this part, beta part, i.e. number 3 part, so on and so on. Now, what do we mean by omega?
1 hour later.
What's up, guys? So just finished class. We're going to be finishing today's part of the experiment by adding the primary antibody. So the main job of the primary antibody is so it can help us actually see what we're looking at when we're actually doing the data analysis part of the experiment, which is going to be conducted next week. Alright guys, so that concludes the procedures and experiments for day number 1. I'll be seeing you guys tomorrow to finish the rest of the experiment, so I'll see you guys then.
The next day.
Hey, what's going on, guys? So today is day number 2 of the experiment. It is Tuesday. We're going to be adding the secondary antibody, so it's a pretty short procedure. So we're going to go ahead and get started. I'll see you guys in the lab. Okay, guys, so in this picture you see the target protein, which means this is a protein that we want to study and do research with. The primary antibody, which you saw me add yesterday, attaches to this target protein. You're going to see me apply the secondary antibody today, which is going to attach to the primary, and on the secondary antibody there's a label that comes with it. So what the label does is that when we go do the data analysis part of the experiment, which we're going to do next week, and we look at it under a microscope, we're able to see where the protein is in the cell, and this allows us to answer various questions. Alright guys, so just got done adding the secondary antibody and finishing up some of my washes. I put the experiment in the environmental control room where it's going to be incubated at 4 degrees Celsius. Tomorrow we're going to be finishing up the experiment, so I'll see you guys tomorrow.
The next day.
Hey, what's going on, guys? Today is Wednesday, which is our last day of our experiments. We're going to be finishing up with a fine dissection, so we're just going to be cleaning up some of the stuff that we dissected on Monday, so that when we look at it and do our data analysis part of the experiment, which is next week, we get a clear picture of what we're looking at. So we're gonna go ahead and do that, and I'll see you guys in the lab. So we're gonna be back on the microscope again to do our fine dissection, which is going to be tweaking up our products that we dissected on Monday. So we're gonna be using this very fine needle to do our fine dissection, and this really allows us to get rid of any unnecessary material that we have on our final products.
Six and a half hours later.
Hey, what's going on, guys? So it is Wednesday night. Finished the experiment. We cleaned up in the lab. Not really much to see there. I'm going to be analyzing those slides that I've been working on next week, and we'll go from there. I'll be making more videos regarding why you should get involved in research, the benefits of research, and why it could help you in your graduate school studies. So with that wraps up my video for the week. I hope you guys enjoyed. Make sure to hit that subscribe button if you haven't already. While you're down there, might as well hit the like button as well, and I will see you guys in the next one.